we are looking to compare ecosystems. This one's a little bit niche and that we're only looking at this particular dot point, but let's break down all the words in this so we know what we're doing. So, very first thing, we are interpreting. QCAA says that basically means we're explaining the meaning or significance of something. We're interpreting ecological data. So, data we could collect might include abiotic factors, biotic factors, and interactions, and all those measures of diversity that we have calculated in the past. We're trying to do that to compare ecosystems, remembering that to QCAA compare means we are finding similarities and differences. And we're doing that across temporal and spatial scales. So this is new information for us. So we have to firstly discuss what these words actually mean. So in this context, a temporal scale refers to a time scale. It can be communicated in years or even lifespan years relative to the generation time of an organism. Gen Y, Gen Z, whatever it is. A spatial scale refers to the geographical area, the physical space, the size, all that kind of thing. So, to compare ecosystems, we collect data, we've done a lot of sampling, we've talked about all these different measures of diversity, and even if bias is completely eliminated and our sampling technique is bang on and impeccable, our sampling is still only representative of a certain moment in time and space. So for example, if you're sampling in the day, we're not going to see those, nocturnal animals, we can't confidently count them. If we're sampling during winter in some ecosystems, you may not see things that are hibernating or, plants that are dormant in the soil. If we sample the shoreline of an aquatic ecosystem, are we actually seeing all the marine life in the Bay Area? Probably not. So all of this can be misleading to our assumptions about a particular ecosystem. To thoroughly compare an ecosystem across temporal and spatial scales, we have to imagine we have the ability to continuously sample. So temporal scale here, we're talking, all right, just start sampling forever. It's all times of the day, all seasons of the year, all decades, whatever. And to do this on a spatial scale, we need to sample a huge variety of areas within a biome. These aren't always practical. So if we look at an ecosystem through the perspective of a spatial scale, we consider size. So an ecosystem exists at a micro, meso, and macro level. The micro encompasses, even though these are quite big, we're talking local areas and biomes. Meso is that middle area. We're talking areas within continents or countries, and the macro is entire countries and entire continents. So that's a pretty big picture right there. And we assume at the bigger picture level that the ecosystems at the micro level are collectively representative of the ecosystem at that macro level. In the same way that if you were like quadrat sampling an ecosystem, the quadrats have to collectively represent the whole ecosystem that you're looking at. So despite the spatial scale we consider, micro, meso, macro, the diversity of a particular ecosystem is going to be similar across all levels. And this is because many measures of diversity are calculated using a ratio of species present in the number of, ratio of species present to the number of individuals in their population. So, it's not just a straight up count. If we talk species interaction, generally the ratio of producers to consumers remains pretty constant, regardless of the level that you're looking at. And since other species interactions work in a similar way, species interactions are similar across all spatial levels as well. And abiotic factors, however, can vary massively from micro to macro. Now, if you considered a tiny, tiny selection, just a tiny little, ecosystem of your own backyard, there's going to be parts which receive a lot of sunlight, a lot of rain. You might have different availability of space in them for species to grow, while other areas have the opposite. So in terms of, terrestrial things, these are things you might look at as well. And abiotic factors are going to vary across that spatial scale. So in Australia, February temperatures might range from low single figures in some places, some places can produce snow weirdly, and then go all the way to the high 40s. So it's pretty rare to find homogeneous abiotic factors at a macro level. So comparisons are usually made at that micro level. We would also consider those things like the soil, the climate, the physical space, the area. And if you're talking marine environments, it'd be all these different things as well. If we look at ecosystems through a lens of a temporal scale, we're talking about change over time. So similarly to our spatial scale, our temporal scales range from small to large, in this case short to long term. So we're talking days, it might be seasonal, or it might be years, decades and centuries. And at a temporal level, biodiversity still is going to differ quite substantially. If we consider when in the day we sample, we have to consider the ebb and flow of things as population numbers change. Some species will die out over time, others will fill in their niche, others will migrate if changes occur in their old habitat. All of these interactions are really heavily influenced by the diversity of the region, which we just said will change significantly over time. In this situation, though, our abiotic factors don't change in ways that are as significant as the other categories. So most abiotic factors may change in the short term, but less so over the long term, say across a year. But really significant change can be seen over a very long term, so that macro level over decades, for example. And to add to this, climate change has increased the rate at which some of these abiotic changes will drift into levels which we aren't used to seeing, so back at that meso level. So if we return to our original statement, we need to be able to interpret our ecological data that we've collected to compare our ecosystems across temporal and spatial scales. So basically we have to do those types of data we've collected, analyze the change if we've continuously sampled an ecosystem over a long span of time or a large physical space. So you're going to need to look at this in a very specific way using actual data probably presented to you in order to draw some conclusions.